but at the beginning I wanted to make a kind of introduction because the workshop is on epigenetics and epigenomics. The hands-on session is more on the ChIPseq analysis on its own, but why? Why is this so important? So first of all, this is a place I work in. This is in Barcelona. It's a part for the Recerca Biomèdica, so it's a research building. We are 7 institutes there. We are in the CRG, and the Center for Genomic Regulation, and I'm very proud of this picture because I took it myself from the 5th floor. It was very early in the morning, as you may see, and it was there. The sun was rising. Everything was super nice, and I'm very proud of the picture. So this is a brief outline of what we're going to do today. So first, I will introduce you the theoretical framework. So what is epigenetics? Why is it important? And so on. And then in the hands-on session, we will explore the data. We will do the basic steps of the ChIPseq analysis. And then we will work basically on data visualization at the genome browser and on some ideas for downstream applications. So what is epigenetics? So epigenetics is difficult to define. It's something, literally, that it's above the genetic sequence, the DNA sequence, right? So as a concept, it has been defined as an epigenetic trait as a stably heritable phenotype that results from changes in chromosomes, in cell behavior, whatever, without alterations in the DNA sequence. So this is what we meant. Epigenetics means it's above the DNA sequence, okay? Something that occurs that makes some changes to the chromatin, to the DNA, but not at the sequence level. So basically, we are talking about the chromatin. Chromatin is this structure that is basically formed by the nucleosomes, this core of histones, and the DNA that is wrapping them. But not only this, there are also non-histone proteins that are binding to DNA. Okay? All in all, this means the chromatin. And the epigenetic changes means what happens at this histone level, non-histone protein DNA level, and so on. So basically, chromatin structure, we all know about it, I hope. So basically, DNA is wrapping these nucleosomes, which is this core of histones, and then it folds different shapes to finally form the chromosome, okay? We have this image of the mitotic chromosome when chromatids are dividing and so on. But this has several layers of folding. And this is thanks to these histone proteins that are able to wrap the DNA sequence and make something that is extremely long, super long, to fit into the cell nucleus, which is tiny. Okay, this is the nucleosome. Very briefly, we know it's a core of histones. It contains histone 2A, histone 2B, histone 3, and histone 4. What happens to histone 1? Histone 1 is outside the nucleosome. It's a linker histone. We're not going to talk about it. But basically, these histones are able to make the DNA to wrap around them, and they contain what we call the histone tails, these things that go out from the nucleosome that are able to be modified. And this is what we work on in our lab with these histone modifications. These histone tails are modified. They can contain different kind of modifications, acetylations, phosphorylation, methylation, etc. And these modifications change between cells. Whereas the sequence of the particular gene is exactly the same in all cells, these modifications that are close in the nucleosomes that are surrounding a particular gene change. And this is actually what we are studying, these nucleosome tails. We have talked about that. So why are we so much interested in these chromatin changes? Well, because chromatin and epigenetics is being related to all processes in eukarya. So chromatin regulation is involved in development, in organism development, in cell differentiation, and in the development of many diseases, of course, because if you are involved in developmental processes, if there is a problem with these epigenetic traits, there is a problem during development, so you can develop some diseases, but not only during development, also during differentiation. If there is a problem during proliferation, for instance, cells overproliferate, they may develop a cancer. So histone modification changes or deregulation actually are related to many types of diseases, to almost all of them, to neural diseases, metabolic ones, cancer, etc. And actually, recently, they have been proposed not as histomodification on its own, but for instance, the resolution is terrible, but here it says plasticity. Plasticity actually means exactly this, changes at the chromatin level. The regulation of plasticity, of changes at the plasticity, have been proposed as hallmarks of cancer. This means that they are actually in the basis of the development of these malignancies. Apart from that, given that they are involved in so many processes, they can also be used to diagnose and prognose such diseases. For instance, in this particular case, these authors, what they have checked is the level of certain chromatin modifications, in particular H3K4 dimethylation. This means, for those that are not very familiar with this nomenclature, this means the dimethylation of the residue 4, the lysine 4 of the histone H3. Okay, we will see this nomenclature all in all because our data contains this kind of nomenclature. And they have seen that different levels of this modification and of H3K18 acetylation can go to can promote different prognosis. So these are survival plots. So certain levels of the modifications indicate better or worse prognosis. So this is another of the reasons why we are so much interested in understanding why histone modifications, chromatin overall, change. And I told you that it's not only chromatin modifications, also other proteins are binding to the DNA. These are, for instance, transcription factors. We know the RNA polymerase machinery is also binding to DNA. And it is also very important to understand where these proteins are binding, okay? Because they are actually regulating, for instance, gene expression. And what makes our cells different probably is the genes that they express. So they perform one or another function because the genes that are being expressed in each cell type are very different. So the binding of transcription factors is also important, and it's also essential to understand how chromatin is folded, is structured, because these factors that bind to DNA are also important to organize the chromatin inside the nucleus, okay? So I hope I have convinced you that chromatin is a super interesting field, and it's essential that we understand the epigenome of all cell types and tissues. So what is the main technology that we use to study the chromatin features? It's basically the ChIPseq, the chromatin immunoprecipitation followed by high-throughput sequencing. I will basically talk about this technology, but it's not the only way we can study epigenetics, epigenomics. We can also look for open chromatin regions. I told you that this, let me go back here, that this chromatin is actually formed by the nucleosomes and the DNA, but the nucleosomes are not equally distributed along the chromatin, so sometimes they leave some gaps. There are some regions where we cannot see the nucleosome, and it's because they are open. They are accessible. In principle, we think that they are accessible because other proteins need to be attached there, so like the transcriptional machinery, and then make their function. So these open regions can be also analyzed with technologies like DNA-seq or ATAC-seq. Okay, so this is something that we can also analyze. Not only this, we could be also interested in understanding how the chromatin is folded within the nucleus. Okay, you may have heard about these high-C technologies, which come from many years ago, that people developed the chromosome conformation capture technologies, in which the goal is to look for which are the closer regions in the nucleus. Because chromatin apparently is not randomly distributed in the nucleus. It has some shape. Some regions tend to be more often close to other regions. They interact. The most common example is enhancer-promoter interactions. We know that enhancers, in principle, need to interact with the promoters to promote their expression at a particular tissue or in a particular moment during a process. But apart from that, these are not the only interactions. There are higher structure chromatin features that can be also analyzed with these technologies. But today, we will focus basically on this technology that I told you, that is the ChIPseq. Okay, ChIPseq is the sequencing of the DNA-protein interaction. So I told you chromatin is very important, transcription factors are very important. So our goal here is to know where our protein is binding to the DNA at the genomic level. Many years ago, people did it at the local level. So we performed experiments, we do qPCRs, and then we check for all our targets. But this was very blind, because if you didn't know the targets of your protein, you could scan the full genome, and maybe the protein in the end was not binding to DNA. So it was a problem. Thanks to the high-throughput technologies, we have been able to do it at the genome-wide level and look for the proper binding sites. So the goal is to uncover these DNA regions, and it has very high resolution compared to other technologies. Also, some years ago, people developed chip-on-chip technologies. The idea is the same, is to understand where my protein of interest is binding at the genome level. But instead of doing by sequencing, they used to do it by array, microarray. You may have seen that you are very young in general, but people used to use these hybridization arrays to check for the binding of their proteins, as they did with the RNA. Also, microarrays of RNA were very common to check for the expression of genes. In the case of the ChIPseq, it is much higher resolution than the chip-on-chip. For obvious reason, in the ChIPseq, you are sequencing all the genome that you are capturing. You will see now how we do that. With the chip-on-chip, we were limited by the sequences that were present in this array, so they were much less efficient. So how does it work? I think it's essential that we understand how these technologies work at the well level to understand what do we need to do afterwards to analyze the data. So what is a chip? Actually, a chip, it's a chromatin immunoprecipitation, literally. What does it mean? We have chromatin, and we precipitate it, we pull it down with antibodies, okay? So basically, what we do, the basal steps are first the cross-linking. This is a fixation step to keep the integrity of our cells, okay? You will see, if you check the literature, there are some people that don't do this step. They do what they call native chips, so that the chromatin is not fixed. It is prone to be disassembled. But in principle, the idea is that you don't get secondary interactions. You get the real strong interactions. It depends on the lab. They prefer one or other methods, but take into account that when you analyze the data, if you have significant differences, it may also due to this step in the protocol, okay? So basically, we can do this initial step, which is kind of optional, and then the cells are broken. We share the DNA, and then we use a specific antibody to pull down the specific regions. Why do we need to share the DNA? Because imagine chromosomes are huge. If you try to put an antibody and immunoprecipitate the full chromosome, it will be impossible. And besides, because we are interested in the enriched regions, which are these regions that are bound by our protein, if we don't cut, imagine that the antibodies were actually able to immunoprecipitate the full chromosome, then we wouldn't have anything enriched. It's important to have these enriched regions. This is the experimental protocol, and then we have to generate, as we have heard in the last lectures, we have to generate our library. So this is an example of the Illumina library also. Most people working on chromatin, I think most of us tend to work with Illumina sequencing because it's fast, it's cheap, and it gives short reads. But for chips, this is not important because our chromatin are actually short fragments because we have actually cut it. Okay, so we don't have long fragments. It's not like long-seq RNA-seq in which we are interested in the longer fragments. This is not the case. Here we don't care because our fragments are actually very short. So Illumina is perfect for these purposes, and it's really very high throughput. We can barcode many different samples in the same lane and get thousands, millions of cells, of reads per sample. So basically what we do, as you all know, is we have our DNA, we have to fix these broken ends, and then we link our adapters, that it's what we're going to attach to the flow cell to make it sequence. So this is there are some people that is working in the lab, they may recognize this figure. This is what a library looks like, okay? This is a profile of a library, and this is actually the size of our fragments by the fluorescence, this means the concentration of our library, okay? So this is what it looks like. You see that the fragments are not very long. The maximum peak is around 300 base pairs. But our actual fragments are shorter because we have added the adapters, remember, okay? We always have the adapters, which are on average 60 base pairs on each side. If it's a normal Illumina adapter, depending on the adapters, they may be slightly different. Or if we have additional barcodes or so. Okay, so our fragments here would be around 170, 180, which is actually the size of the DNA wrap in the nucleosome. Okay, and with that, we go to the sequencer. And this is the process. So I've been very fast. Cool. The bioinformatics analysis, which are the main steps? Okay. We will see most of them more carefully. I will go quite fast because I think most people are actually very much familiar with these mapping processes, but I would like to mention some particular ideas about mapping this ChIPseq data. Basically, the first step is to align our sequencing reads to the genome reference. Make the peak calling. What is the peak calling? The peak calling is to basically understand, to ask the program, which are these enriched regions. So we have a particular region that shows a lot of reads. This means that it has been enriched. We need to retrieve the coordinates for these regions, and this is what the peak caller actually does. And then there are some quality control steps. We will not touch them very much. We will see some correlations and so on, but we will not talk very much about them because I would like to check more about the visualization and other analysis. Okay? So for the bioinformatics analysis, the first step, we said it's read mapping. There are many programs that have been developed to do this task, okay? Basically, we need, as you know, the root sequences. This is FASTQ files that come from the sequencer, and then each read is aligned to a specific region in the genome. There are more than these pipelines. One of the things that we may think of, it's actually this that we are seeing here. I'm not sure if many of you have ever thought of it, but when you sequence a read, the read we said it was like 300 base pairs, okay, counting with adapters, but the actual region of interest is 180. What you're sequencing with Illumina is 50 base pairs. So this means that you sequence just the ends. We don't sequence in the middle part. But what usually happens is that your protein is actually falling in the middle part of the read because it's protecting the region for the digestion. So what usually happens is that you get the reads of the extremes. Your Illumina reads correspond to the extremes of the actual site. Okay? So when you plot them, you can see that they are actually not, they leave like a gap in the middle. So some of these mappers, what they do is they have functions that allow you to merge these reads. Okay, so you can have a parameter like extension or so, by which you say, okay, my read is 50 base pairs, okay, in each side. But I know that my fragment length was 180. So I just tell the program, okay, extend it. And it does. So, or I can extend it or I can move them. Both options are possible. And then in the end, what you have, it's a peak in the proper region, not two peaks neighboring the region.
Does it mean that you always use like paired end sequencing while you're seeing
No. Actually, we don't. But this, you can do that. You can use parent. But in our case, we don't do that. But the thing is that even when you don't do parent, as the protein of interest usually is protecting the nucleosome in particular, it's like protecting the DNA from breaking. So what happens is that if you imagine the nucleosome, you have the two tails. What you're capturing is the two tails. But the nucleosome itself is in the middle. So you see, maybe it's even more evident with transcription factors, that maybe if they are really binding DNA in a strong manner, they protect a lot the region. So it's very common that you see when you check the reads one by one. If you do like if you see the BAM files, we can actually check. you check the BAM files, you have the two sides, and you see that they actually leave like a gap in the middle because they tend to be protected. So it's very common, even being just single end. So
what happens if you have sequencing with long-read sequencing? Like
for the ChIPseq, I wouldn't recommend to do, well, long. The thing is that how long can it be? If your fragments are, the maximum in our library was about 500. Yeah, if you have if you do long-read sequencing, you will have the full read. Then you don't need to extend, obviously. But they cannot be very, very long because your fragments are actually very short. And people don't tend to use long-read sequencing for this kind of analysis. Yeah. So this is one of the things I wanted to mention about the mapping. Then the second step will be the peak analysis, the peak calling, sorry. Again, there are plenty of pipelines that can do this work. Just one thing more about the mapping. This is a problem that has been more or less solved because sequencing exists since many years ago, and many people have been working on that. But still, today there are discussions on how alignments have to be performed, especially at the chromatin level. Most of you have probably worked already with RNA-seq, which generally, I mean, for instance, the STAR was developed to map RNA-seq in particular because it has this split function that allows you to take a part of a read in one exon, part of the read in another exon, and identify it as the same transcript. It's very good. But RNA-seq in general takes into account, well, general no, it is about genic regions. And genic regions can be repetitive, more or less, but they are quite unique. In the case of chromatin, this is not the case. We have huge amount of DNA that are actually very repetitive. For instance, telomeres and centromeres. This is something that we have to take into account when we map ChIPseq data. Not only ChIPseq, any kind of DNA data. So this is something that we will also see, but there are some parameters that we can set for these multi-mapping steps that sometimes we just do by default. But we have to take into account that these parameters may need to be different depending, for instance, on the proteins that you are chipping, your immunoprecipitative. Because if your protein is a euchromatin protein, so something that falls in non-repetitive regions, that's perfect. But if your protein is actually marking repetitive regions, this is actually an issue. So something to take also into account. We were talking about the peak colors. So in the case of the peak colors, this is still, I mean, it's also, many people have been working on them. There are many pipelines so far, but still, some people think that there is room for improvement because you will see when we analyze our data that it's still not perfect in some cases. So our eye is more efficient in defining the peaks probably than the peak colors. And this is because at the computer level, it's very difficult to differentiate enrichment from not so much enriched regions. Basically, these peaks represent regions that are enriched. So this is an example, and we will do things like that, okay? So these are our peaks, these are our enriched regions, meaning that our protein is basically, the genome is sitting up here, and then we have been able to retrieve, to capture these sequences with our ChIP technology. And what the peak color will do is to tell us, okay, this that is very evident by the eye. What are actually the coordinates of this in the genome? This is what we need. Because it's very nice to see it, but what we need to be able to manipulate, to work with the data, is actually the region. Where is it in the genome? Then, quality controls. As always, controls are essential. We will see that we have many possible controls here in the ChIPseq. We can also not to use controls. We will talk also a bit about that. But basically, these are controls of the technology, of how the experiment has worked, okay? These are, if you want to explore more, there is a lot of information in the ENCODE ChIPseq guidelines. Actually, the ENCODE project, as you may know, has worked for 20 years on developing technologies and computational methods to analyze this kind of data. So they have put also a lot of effort on developing this kind of metrics to understand if experiment is working or not. Because at a certain level, we need to know if our experiment is actually good enough to be to perform the downstream analysis. Okay, so basically, they have implemented these two metrics. The non-redundant fraction. The non-redundant fraction is a metric to identify which is how variable our library is. What does it mean? Again, for RNA-seq people, libraries on RNA-seq tend to be super variable because there are many RNA-seq molecules in the cell, in the tissue, in the whatever thing you're working on. There are many. And when you fragment the RNA to do the typical RNA-seq, you have many different fragments. So when you do a library, the library is super diverse. Actually, we will see when you check for duplicates in the RNA-seq level after sequencing, there are not, in general, if the library is properly performed, there are not many duplicates. This is not the case for ChIPseq. You usually have many duplicates. And this is because the technology starts from a tiny amount of DNA, and you immunoprecipitate it, which means that you end up with really tiny amount of DNA, which has to be amplified. So you can get a lot of PCR duplicates. And this is what actually this matrix is telling us, okay? So you have a low complexity library, which is this case. It's a chip that has worked, kind of. You have a lot of duplicates. And this is what happens. When doing the PCR, you have many reads that are exactly the same. They are identical. This is low complexity. In a good experiment, what you should have is many reads that are different because they come from different cells. Because each cell has just 2 copies of the same DNA molecule, of the same sequence, okay, of the same gene. But these come from many different cells. So in the end, what you have is different fragments of different length covering all this region. And the non-redundant fraction, actually what is taken is this proportion of duplicates versus non-duplicates, duplicated reads. And this is the first metrics. The second one, it's a FRIP. FRIP stands for fragments in peaks. It is what it is. So remember, we have our chip, and then we want to know if the experiment is good enough or not. Remember, for instance, in this case, okay, we have these peaks here. What FRIP means is that how many of the reads in my sample, in my sequencing sample, are actually falling in my call peak. So in this case, in the case of the FRIP, we need the peak caller part, okay? Because we need to know where our peaks are being called, where the program thinks that our regions are enriched, okay? So, and then when we have these regions, we ask it how many of the reads in our sample, which is the proportion of reads that fall within these peaks. This will give us a rate ratio of the real peaks versus the background, okay? The higher the FRIP, the better, okay? Because this will mean that you have really enriched for specific regions and that your background is super, super, super low, okay? What happens in this case is very clear. I see the peaks very clear. You will see in the examples that we have that it is not that clear always. So sometimes we have very high background, or our protein of interest is actually very, spanning in a broad manner in the genome, so it's difficult to distinguish between the peak and the background. So it is not that easy. So ENCODE has also set up different metrics depending on the protein of interest and on the particular, or in the histone that you are checking. For instance, transcription factors are far more difficult to immunoprecipitate, so the tolerance to these metrics are slightly different, because also they are very peaky, meaning that you don't have long regions that are covered by the transcription factors, and then many times happens that you don't have so many reads falling within your peaks. You have many more reads at the background, even if the chips has worked. So this is another thing to take into account to check for the experiment. One is done, the NRF can be checked at the just with the duplicates, and in the case of the FRIP, we need also the peaks. Okay? And just before we go for the hands-on session. What is the epigenetics analysis on its own? Okay, so we have our data, we have mapped and we have mapped it, and we have checked for the peaks. And then what can we do with this data? We can do many things. Very important for me, and we will spend some time on that, because I'll tell you later, but I'm from the experimental lab, and then I moved to computational. But from the experimental lab, it's very important to understand if things are working in a visual manner also. So we spend a lot of time also trying to visualize our data. And in this case, you see, browsers. In my particular case, I love the UCSC genome browser. We will talk about it later. But there are other browsers that help a lot on visualizing this data. Because sometimes you start with some numbers, we start with the reads, we get the peaks, and we never look at the data on its own. And when you eventually do that, you realize that maybe the data was not as good as you expected, that everything was a bit random, and then it's very shock. It's a shock when you realize all these things. So it's very important to start looking at the data from the beginning and then analyze all the data. Actually, sometimes maybe it's not even necessary to analyze the data, depending on what we see. We will also check. This is something that is very important for us. And then some other analysis that we can do. We can use, we can obtain the target gene. So I have a transcription factor that is binding somewhere in the genome. I do my experiment. I obtain the list of target genes, if I can, because who knows if the transcription factor is actually binding the transcription start site or enhancer regions or whatever. So we can also check which is the type of regulatory region, or if it's binding to heterochromatin or whatever. So we can answer what is my protein binding, what are the target genes. Once we have a list of target genes, we can also check which is the gene ontology function of these genes. Okay? So what are these genes doing? I'm working with a transcription factor that is basically expressed during development, but maybe it's not related to the developmental process. Maybe it's a metabolic factor that needs to be expressed in a particular moment during development. This kind of analysis will answer these questions. And other questions that we can answer is how does chromatin change along dynamic processes, for instance? Because this is actually the key point of studying chromatin, as we were seeing at the beginning. We want to know what is chromatin doing and what are the phenotypes associated to changes at the chromatin level, either natural phenotypes like differentiation or so, or diseases, etc. And in this sense, I wanted to show you a couple of examples about what we can do when we analyze chromatin. This is, for sure you have seen this kind of plot. This is ChromaHMM. It's a very famous pipeline. It's a very famous program to segment the chromatin. What does it mean, chromatin segmentation? What the idea here is to, doing a dimensionality reduction. So we want to reduce the amount of data. We have the case of the ENCODE. They usually work with 5 or 6 different histomodifications. You will see in our case we have 9. So this is a lot of information. We need to somehow reduce this information to get which is the chromatin profile at each particular genomic region. So what they developed these hidden Markov models that allow you to segment the chromatin. So it's basically making bins of each particular region. And then it takes all the input files that you give it, okay? And it's telling you, okay, this particular region has these marks here. This will be, I don't know, I'm not sure what it is now, but this may be enhancers or enhancer regions. These regions here, they don't have anything, heterochromatin. This has HCK4 it's a promoter, and so on. So depending on the combination of histomarks, it will give one result or another. And actually, this is what we see here, okay? They are using, in this case, 6 histone modifications, and they define up to 18 states. Each state is actually a combination of histone modifications in this case. That's it. And you can see here, these first states here are characterized by high HCK4 trimethylation and some acetylation in this case. In this case, It's also acetylation and monomethylation and so on. And all of them are different. They usually also have absent chromatin states that they differ in, here it is not very clear, but usually they differ in where in the genome they are. So you can have the states as combination of marks, and then you can check where these states are, if they are close to the transcription site, if they are in exons, if they are intergenic, whatever, and what is, how they look overall. This is another approach, very similar, which is the chromatin colors. Probably many of you have not heard about that. I don't know. This is a very nice paper in Cell some years ago. But the idea was exactly the same, but it was like reducing even more because these people end up just with 5 colors. This is Drosophila. It was done using tons of data of transcription factors and chromatin modifications on Drosophila. And then they defined these 5 colors, which in the end mean the same. So their black chromatin represented regions that were not bound by anything in the genome. They couldn't find anything in Drosophila. And then blue regions were facultative heterochromatin. And red and yellow were actually active regions, transcriptionally active or transcription start sites or whatever. And green were constitutive heterochromatin. So these are a couple of ideas. And people working in chromatin, many times, they try to go into that direction. Because actually it's a way of reducing a bit the amount of information. Because when you have so many chromatin modifications, so many cell types, and so on, it's actually hard to integrate. In our case, just very briefly, I'll show you one example, where just very small example of some of the things that we are doing in the lab. We are actually working with a human model. We work with different models. In this case, it's human. It's a human transdifferentiation model. This is a cell line that is called BLAIR1 that was developed in our institute also, that is able to transdifferentiate. It's a privy cell, so it's in the lymphoid branch, and these cells are able to transdifferentiate into macrophages. Okay. For those of you who know a bit about blood, privy cell is lymphoid, which is one branch, and macrophages are myeloid, which are very far. Okay. So naturally, in a natural manner, the cells don't do that. They cannot do that. But by adding this particular transcription factor, that is CBP alpha, these cells are forced to make this change. And then this, we think, occurs with strong changes, with dramatic changes at the transcriptomic and epigenetic level. Okay? So this is a process that takes up to 7 days. And actually, after 7 days, these cells are fully differentiated, and they are even functional. It is described that they are even to phagocyte. Macrophages, they have this immune function, and they are even able to phagocyte. So what we have done is actually to collect cells at different steps during the transdifferentiation. So we have collected these cells at 12 different time points. As you see, we have cells. We have collected very tight time points at the beginning of the process because we expected many of the changes to occur just at the induction of the transcription factor and a bit less sparse at the end of the process. Okay? So we have whole RNA-seq data, and also 9 different histomodifications covering all the regulatory regions that we were interested in, because we knew by the literature that they were actually, in principle, deposited in different places. For instance, K4 trimethylation is basically at the promoter levels, at the transcription start site, not at the promoter. And then monomethylation, K27 acetylation, and K9 acetylation tend to be at the promoters and enhancers, as well as K4 dimethylation, actually. Depending on if they are there or not, they have been assigned different roles. For instance, K27 acetylation at enhancer regions is associated to activity of the enhancer. Whereas if we just find the K4 monomethylation, in principle, it's thought to be just prone to be activated, but not functional. And we have also gene body histomodifications. These are modifications that are basically deposited within the gene, as it was in the literature. And then also heterochromatin proteins, heterochromatin modifications, the K27 and K9. So what we did was to, just very briefly, to do a similar approach to this chromHMM, but in our case, we had an additional variable, which was the time. Okay, remember that we have 9 histone modifications, but along 12 time points. So we took advantage of this variable to do another thing, to do a temporal segmentation. So we are not segmenting the genome in pieces. So in general, these methods, what they do is they take the full genome and they tell us this is enhancer, this is black, whatever thing, okay? What we are doing is to do that, but a long time, okay? So what we get is transitions between one state, between one time point and the next. Usually, chromHMM, what gives you is transitions between one segment and the next segment. So what is more probable to have? A enhancer region that the next region will be a transcription start site. In our case, this is not what we're doing. We are checking which are the changes a long time. So, and we were checking that at the gene level. So it means that we have not incorporated all the data in the genome, but just the genomic level, because the gene level, because we were interested in the relationship between these chromatin modification combinations and the status of the transcriptional level. Okay? So in the end, we end up with 5 different states compared to the 18 states that were defined before at the genomic level. And these states, again, are defined by different histone modifications. Okay? So we have this strong state that has almost all our histone modifications, but the K27 trie, an active state that contains almost all of them, but K9 trie and K27 trie, and so on. Okay? We have a bivalent state. We don't know, well, people not familiar with chromatin will not know, but bivalent means that you have both activation and repression marks. Activation being K4 trimethylation, and repression being the 9 and 27 trimethylation. They, in principle, shouldn't be in the same regions, but there are some genes that present both marks in parallel, and this is why they are called bivalent. And then we have also one state that we call low, that is very low marked in general, but it has some K4 monodie, meaning probably it's furon enhancers, and another state that is actually absent. It has nothing. Just as a curiosity, I told you that these 2 chromatin marks, K9 trie and K27 trie, are actually repressive marks. They are found in heterochromatin, so in non-active regions. But as you see, our strong model, our strong state, also contains K9 trie. And this is one of the unexpected things that we think are very interesting, because actually we didn't expect this modification in particular to appear here, and we have explored it a bit more. And actually, many genes that are active during transdifferentiation contain this modification. So, as I told you, chromatin is a big field, and it changes depending on the model that you are looking at. So it's very nice to go on with it. So what we did with this temporal segmentation is, as I told you, we had the 12 time points and all the states. So we check how they were looking. They were changing along the transdifferentiation. So we have per each gene, one state at each time point. So we just plot all genes at the 12 time points. And then we check how they change. And to our surprise, they were basically not changing much. They are quite stable. Okay, so for instance, these are silent genes. Okay, these are in purple, which means that they are absent. This is what we more or less expected, because if you are not expressed, it may happen that you have some repression marks or you don't have anything. That's okay. But then here in light gray, it's not very clear, but some of these bars are actually differential expressed or stably expressed. Obviously, they are, we expected them to be stable also at the level of chromatin. But what surprised us was the differential expression. So many of these genes are actually differential expressed, meaning that they change from the beginning to the end of the process. Okay? But still, we saw a lot of stability. You see all this black, all these yellow bunch of genes here. They are active always, independent of the changes at the chromatin, the transcriptional level. And the same for these strong genes here. Some of them change from active to strong or from bivalent to strong, and so on. And actually, even some of them were located in the low marking state. So, and they didn't change, even though their expression was changing. So this was actually very surprising to us because we expected many more chromatin changes. But this was not actually the case. It's basically transcriptional changes. And with that, well, this is what I was telling you about the transition. Basically, we take the transitions from our temporal HMM models to identify which are the most abundant changes between time points. So the most abundant changes actually are from low chromatin, low state, to strong or to active, and the other way around. And actually, they coincide with expected changes at the transcriptional level. So if you go from a lower marking level to a higher marking level, you tend to also express, to show a higher expression of the genes and the other way around. So if you go from high marking to low marking, you tend to be downregulated. But still, there are not many changes, as you see there, okay? And the consequence of that is that actually when you check at the correlations, at the time correlations, they are actually much lower than expected. This is my last slide about that. So here what we plot is the time course correlations compared to the individual correlations. Individual meaning the same time point between chromatin and expression. So this is what we usually do when we compute correlations between chromatin and expression. We take the cells, we have the matrix with the values of expression and the matrix with our values of the chromatin. We compare and we have very high correlations. But if we, instead of doing that, we do it across time. So we check which is the correlation along the 12 time points. What we see is actually much lower correlations. For instance, this is the K4 trimethylation. It's a typical mark for the transcription start site. And you see at the individual level, each time point has very high correlations. Perfect. But when you check across time, the genes that change expression, they correlate very low. I mean, this is like 0.4, which is not very strong. Okay? So this is actually the consequence of this stability at the chromatin level that we didn't expect and that we couldn't have checked. We wouldn't have been able to see this phenotype without this dynamic process. Because usually what we are used to do is to take particular cell type and perform all the experiments on the cell type. But in this case, being so tight, being so many cells and so many time points have helped us to understand that actually the process is not that well correlated, that chromatin and transcription don't need to go in parallel necessarily. Just someone is more prone to chromatin. I told you that we had some genes that also had K9 trimethylation, which is the mark. It's a repressive mark in principle. And when we check the correlation across time, we see actually that the correlation is positive. Slightly positive. It's not very positive, that's true. But it is positive. So that it doesn't correlate at all with repression, but the other way around. So this is also another of the striking results that we got, and that we were able to observe because of this dynamic process, of the amount of experiments we perform.